I'm particularly excited about the opportunity to be able to communicate some of our research results to folks who are actively involved in designing and implementing invasive management techniques. One thing I wanted to point out before is that in the course of this work, we actually weren't able to remove Multiflora rose as part of our experiment. So if anyone has ongoing management efforts targeted at this species and you'd be interested in collaborating to look at tick-borne disease risk, please get in touch. I'll have my email up on the last slide at the end of the presentation. So first, I just wanted to acknowledge all my collaborators on this work. We had Greg Shriver, Jeff Bueller, and Jake Bowman at University of Delaware, Beth D'Amico at the U.S. Forest Service, Dustin Brisson at University of Pennsylvania, and Andreas Kogard with the Centers for Disease Control and Prevention. And along similar lines, I had also people and funding sources that made all of this work possible. So back to the question, does Multiflora rose invasion increase Lyme disease risk? Well, I'm going to do something a little bit unconventional today and give you the answer up front, but you'll see it's not quite as straightforward as the question might imply. And basically the answer is it does increase Lyme disease risk at fine scales, but at broader scales, probably not. And as you'll see, this is because rose invasion seems to be associated with specific habitat changes that might actually make habitat less suitable for ticks. I hope that this perspective will especially be valuable for managers who are weighing lots of different priorities and goals when it comes to invasive species management. So being in New York, unfortunately, I'm sure most of you are familiar with Multiflora rose. Just some brief background, it was originally brought over from East Asia as a rootstock for ornamental roses, and later its planting was actually encouraged as a means to control soil erosion and provide a living fence for cattle. It's turned out to be a pretty aggressive forest invader. In the mid-Atlantic, in Delaware in particular, it's one of our most common understory forest invaders, particularly in small forest fragments. In some cases, we see more than 60% of the total understory cover as just basically a monoculture of Multiflora rose. And we've heard a lot of anecdotal evidence about links between Multiflora rose and tick-borne disease, but our study is the first to directly address or try to quantify what those effects might be. So when we talk about tick-borne disease risk, we're really talking about these two factors shown here, tick abundance and infection prevalence. Tick-borne diseases like Lyme disease are transmitted among vertebrate hosts through the bite of an infected tick. So in order to increase disease risk, we need to see increases in one or both of these factors. So simply having a whole lot of ticks out there won't necessarily make disease risk very high unless at least a certain portion of them are infected. And likewise, we could have a 100% infection rate, but if there are only 2 ticks, disease risk won't be very high either. So here we're interested in looking at how Multiflora rose may affect one or both of these parameters and ultimately impact disease risk. Most of the work, most of the prior work, maybe some of you are familiar with, which has been done looking at invasive shrub effects on tick-borne disease risk. Most of this work has really focused on 2 invasive shrub species, Amur honeysuckle and Japanese barberry. And at fine scales, both of these shrubs have been shown to increase this tick-borne disease risk, but through different mechanisms. I'll give you a brief overview of kind of what we know about the impacts of these 2 shrubs at fine scales. The ticks are obligate blood-feeding parasites, and they have a pretty slow life history. So their survival and population growth is largely regulated by host availability. They need hosts to feed on, and also microclimate conditions, because they spend the majority of their 2-year life cycle actually off of hosts, just trying to survive essentially on the ground. So Amur honeysuckle has been shown to be used preferentially by white-tailed deer, which we know are important hosts for tick reproduction. So as a result, this preferential use by deer of honeysuckle has caused us to see greater densities of ticks within stands of honeysuckle compared to adjacent areas outside of it. Japanese barberry, on the other hand, seems to increase tick abundance through direct effects on tick habitat. It has this dense growth structure, which has been shown to create a more favorable microclimate for tick survival. That dense growth structure really keeps temperatures more stable and relative humidity higher, both of which are good for ticks. So that's what we know about these shrubs and tick abundance, but then there's also the pathogen prevalence component of disease risk. Amur honeysuckle actually has not been shown to increase infection prevalence among the ticks within it. It's just that the overall density of ticks is so much higher within honeysuckle that disease risk is greater there. Japanese barberry, on the other hand, has been shown to not only increase the abundance of ticks within it, but also increase the proportion of ticks which are infected. And we think this is because barberry creates great habitat for infectious host species like white-footed mice. And so when we have aggregations of ticks and infectious hosts, then we get higher overall pathogen prevalence within the tick population there. But so far, most of this work has been done at fine scales, and it's still pretty unclear whether these are just associations between invasive shrubs, ticks, and pathogens, or whether they might scale up to broader, more regional scales to affect tick range expansion and increase human disease risk over really large areas. So we already have lots of good reasons for removing invasive species due to the harm that they cause surrounding ecosystems, but we wanted to know really, can we add human health risks to that list, and what role are invasives really playing in both range expansion and rising incidence of these diseases. So in this study, we looked at how Rosa invasion affects disease risk through parameters, tick abundance, and infection prevalence. And we wanted to look at multiple spatial scales. So we focused on 2 spatial scales here. The first one being similar to the prior work that's been done. So looking within forests that already have a significant amount of Rosa invasion and comparing tick abundance and pathogen prevalence within stands of rose compared to just outside of it, but within the same forest patch. And given rose's many similarities to honeysuckle and barberry, we expected it might have similar effects at this scale. And then we also looked at a broader scale to compare forests with and without significant amount of rose invasion. So we conducted all this work in a network of forest fragments centered around New York. These forest fragments are part of a long-term ecological research and monitoring program that was established cooperatively between the University of Delaware and the U.S. Forest Service. But they provide a great site for this type of study because we have a gradient of multiflora rose invasion, and this area is also known as kind of a hotspot for Lyme disease and other tick-borne diseases. So we used standard carbon dioxide baited traps to test our hypotheses at these 2 different spatial scales. Within rose invaded sites, we set up traps in a paired design, So we deployed one trap in a location within Multiflora rows and then set up a pair 25 meters away in a random cardinal direction, with the only criterion being that it couldn't be within Multiflora rows. And then within the uninvaded sites, we just simply chose 4 random locations where we did our trapping. We repeated this effort for 2 seasons, and I'd also just like to point out that we're really focused here on the nymph stage of black-legged ticks, since that's what's considered most dangerous in terms of Lyme disease risk for humans. So I'll jump right into our results here in terms of tick abundance. At this fine scale within invaded forests, our results were consistent with prior studies on other invasive shrubs. So we found roughly double the number of ticks within stands of rows compared to their paired traps just 25 meters away outside of rows. However, unexpectedly at this broader scale, we actually found the opposite effect, and ticks were way more abundant in the uninvaded forest fragments compared to the rows invaded forest. So to understand what was going on here, we went out and collected a whole suite of habitat and landscape variables that we expected might affect tick abundance at this scale. And then we used a couple different modeling approaches to both rank those variables in terms of their influence and importance, and also to look at the exact nature of the relationship between those variables. So here I'm going to show you the results of our best model for tick abundance, and you'll see log tick abundance is on the y-axis of all 4 plots, and then on the x-axis you'll see just these 4 most influential predictor variables that came out in our best model. So starting at the top left, we had leaf litter volume, distance to nearest stream, distance to nearest road, and the average diameter of breast height of the dominant canopy tree. I'm happy to talk more about this later if you have questions about specific variables here. But for now, I'm really going to focus on what was far and away our most influential variable, which was leaf litter volume. So to understand how this whole story fits together, we actually need to introduce another exotic taxa, which are Eurasian earthworms. So when earthworms invade a patch of forest, they devour the leaf litter, which we know is bad for native plants, and may even help facilitate non-native plant invasion. And this is something we observe as a patch level effect within our study system. Those Multiflora rose invasion also have lots of invasive earthworms and very little leaf litter on the ground. And this loss of litter, of course, affects litter arthropods. And in all of our efforts to control ticks, we often, I think, like to forget that after all, they are native litter arthropods that are vulnerable to these reductions in leaf litter just like anything else. And this is nothing new. It's been well documented that removal of leaf litter results in subsequent declines in tick densities. More recently, Burtis et al. Even linked the impact of invasive worms to leaf litter losses and subsequent reductions in tick densities. But how do invasive plants fit into all this? So if you remember, this is a patch scale effect in our system. So Multiflora rose, lots of Multiflora rose invasion exists in patches with lots of earthworms and almost no leaf litter. And that lack of leaf litter creates a pretty hostile environment for ticks. But these invasive plants like Multiflora rose have a dense growth structure which may improve microclimatic conditions and encourage use by hosts that are important to tick reproduction and survival. So we think that invasive plants like rose at this fine scale are probably providing refuges for ticks in what's otherwise a pretty hostile environment without that leaf litter. So that's why when we look at fine scales, it seems that ticks are super abundant in invasive plants like rose. However, uninvaded forest fragments with intact litter layers are probably providing much more suitable habitat for ticks. So to sum up this aspect of the study for tick abundance, basically we do see a positive association between rose and ticks at a fine scale, but the effect is reversed at the patch scale, comparing invaded and uninvaded forest fragments. And this has important implications for management and the need to consider what types of forests are under your management purview and where tick control efforts might be best directed. Invasive plant removal might still be a valuable strategy if there's a significant amount of invasion to deal with, but in terms of overall tick densities, it seems that they're a lot higher in forests that have that nice deep litter layer still. So now we know about one half of the disease risk story, but what about infection prevalence? So infection prevalence in a given tick population results from the interaction between ticks and a community of host species. We know that host species vary in their ability to carry and transmit pathogens to feeding ticks. So ultimately, which host species ticks feed on impacts the overall level of infection prevalence in the population. So we wanted to understand again how multiflora rose invasion affects pathogen prevalence at these two different spatial scales. So we used the same ticks that we collected and the same design from the first half of this experiment, and we tested them for 4 different pathogens known to cause disease in humans. We're largely focused here on Borrelia burgdorferi, which causes Lyme disease, but I'm going to present all the pathogen results here just so you have an idea of their relative prevalence. Let's take a look at what that looks like. The good news is roughly 85% of all the ticks that we tested were negative for all 4 pathogens. Now we'll take a look at the breakdown of the pathogen-positive ticks, most of which were positive for Borrelia burgdorferi, which causes Lyme. We had 1 tick that was co-infected with Borrelia and Anaplasma. Roughly 2% of ticks overall were infected with Anaplasma. We had a few with Borrelia miyamotoi, which causes a relapsing fever condition. And we didn't detect any Babesia microti in any of the ticks. So for me, I'm really going to be focusing on Borrelia burgdorferi disease. So to get back to our questions at these 2 different spatial scales, basically we started by asking, at a fine scale, what's the probability of any tick collected from within multiflora rose of carrying Borrelia? And how does that compare to a tick collected from just outside of rose, but within the same forest patch? And the answer is they don't differ. When we look at pathogen prevalence at this fine scale, we did not detect any significant difference among ticks collected within and just outside of multiflora rose. And this result is consistent with what has been shown for honeysuckle, where there's no difference in pathogen prevalence at this fine scale. But when we look at a broader scale, we see quite a significant difference between invaded and uninvaded forest fragments, with invaded fragments having roughly double the pathogen prevalence compared to uninvaded fragments. So what's going on here? I wanted to show you some examples of what these sites look like. In our landscape anyway, we have kind of 2 extremes in terms of invaded and uninvaded fragments. Our invaded fragments are quite a bit younger and have this really dense understory structure because of the invasion. The uninvaded fragments, on the other hand, they don't have many invasive species, but because of overbrowsing and probably some other below-ground processes, there's almost no native understory left at all. So as you can imagine, the wildlife communities inhabiting these different types of forests are probably quite a bit different. And we expect that pathogen prevalence is higher in the invaded patches because overall ticks within those are feeding more on infectious hosts like white-footed mice, which are going to enjoy living in a site with lots of dense understory structure. So again, back to this original question about how multiflora rose invasion affects Lyme disease risk. The answer to this really depends on your scale of observation. So when we take these two parameters together to understand disease risk, we see that at fine scales within invaded fragments, you are more likely to encounter an infected tick within stands of rows compared to just outside of it. Our studies have shown that removal of honeysuckle and barberry help reduce disease risk, and we would predict that rose removal would have a similar effect at that scale. However, at the patch scale, both of these habitats are degraded, and both are probably bad in terms of disease risk. The invaded fragments have greater pathogen prevalence, while the uninvaded fragments with those thick litter layers have greater tick abundance. Overall, we expect that the uninvaded fragments may actually be worse in terms of disease risk, because although pathogen prevalence is lower, there are so many more ticks in those uninvaded fragments that overall your chances of encountering an infected tick are going to be higher in the uninvaded fragments. So we have lots of exciting directions we'd like to go from here, including manipulating rows, removing it, restoring native understory structure, combining these kinds of things with deer management efforts. And I'd love to talk with any of you who are interested more about all that. But thanks again for your attention, and I'd be happy to take any questions here right now.